Okay, so I'm going to do a quick review then. We know that our immune system basically works on, understand the methodology. The principle is stop a pathogen from entering the body. That's the first thing. Not that different from our country's security system. Stop an enemy from entering. Put things so that, radar, etc., so that enemies can enter. If they do enter, then have some way of taking care of these enemies without causing too much of haywire. So basically, I don't know why this keeps telling me this Microsoft search. Anyway, so immune system, we just learned that there are two forms of immunity, two components, overarching two components. And those two are innate and adaptive, acquired or adaptive. So innate, adaptive. Innate meaning you already have certain barriers, certain cells, certain proteins that you are born with, and they are going to keep working to protect our bodies. Adaptive is the other. So think about, we have some built-in system along our country's borders. You got wall and, controversial or not, I'm just using the example. We got wall, we got barbed wires, and we got radar system, we got checkposts. These are all basically, think of them, these are, a country comes with this. Adaptive is when you have a novel enemy which has been able to penetrate into within your borders, and you know that that's an enemy. And basically, before you respond to that enemy, you have to learn about who's that enemy, right? Or think about it in your house. You got a pest. We got things in place so that rats can't enter, pests can't enter. And that's basically part of innate immunity. You got things in place so that microbes or pathogens cannot enter. And if they do enter, they will be taken care of fairly quickly. Adaptive immunity is basically, there is a pathogen. Learn about what that pathogen is, meaning learn about its protein. It's all about protein, right? Learn about the pathogenic protein and come up with a counter protein, or come up with a cell that can recognize this protein and it can destroy that pathogen. So basically learn, produce some things that you did not have before, and take care of this pathogen here and now. Now, going forward, keep a memory of what we did here so that this pathogen shows up again. We can handle this pathogen in the same manner that we did now. So that's why it's called adaptive or acquired, meaning you acquire this immunity as you go through life and become exposed to various types of pathogens. This is why we say adaptive immunity has memory and discrimination between self and non-self. Innate immunity has some discrimination between self and non-self. I mean, that's how NK cells and phagocytes would work, but it has zero memory, meaning there is nothing novel that would happen. Nothing new, no new cell will appear, no new protein will appear that wasn't there in innate immunity. In result of adaptive immunity is a new colony of cells, almost new. You must have one at least to begin with. And new forms of proteins like antibodies will appear in your plasma that weren't there before, which is why it's adaptive. Also keep in mind, innate immunity, often the goal of the innate immunity is to keep the infection pathogen in check until adaptive immunity can take over. So ultimately it's the adaptive immunity that basically will take over and kill off the infection. Innate immunity often, it's just by a few days, because adaptive immunity will take a few days to learn about the pathogen and be activated. Until then, keep the infection checked by innate immunity. So, but innate immunity also, there are components that will serve as barriers. So as we know, mechanical, chemical barriers, these are stop the entry of the pathogen into your internal system. When you say internal, internal, inside, which literally means interstitial fluid. Think about it. If a pathogen comes through your skin, and let's say it penetrates through the 30 layers of dead cell, where does it end up being? Interstitial fluid. If a pathogen is able to penetrate through the mucous membrane, which is lining all of our open cavities, where is it going to end up? Interstitial fluid. So the internal environment of the body is basically interstitial fluid. So the barriers, mechanical, chemical barriers, are to stop the pathogen from landing here. This is our internal environment. Now, if a pathogen does end up in that internal environment, well, then what do? Well, internally, we got cells, we got proteins. Internal, I won't say barrier, but internal agents, barriers, whatever you choose to call. We got proteins, antimicrobial proteins, antimicrobial cells. These proteins are interferon and complement protein. The cells are NK cells and phagocytes. Interferon complement proteins, NK cells, and phagocytes. Okay. So as we know, interferon is for what type of pathogen? What type of pathogen is stopped because of the action of interferon?
Virus.
Virus. And interferon is secreted by? I'm going to start over. Interferon is secreted by? Who secretes interferon? A nucleated cell that has been infected with a virus. Viruses don't go after cells like RBCs, because viruses' goal is basically manipulate your our cells, genetic machinery, and produce their offsprings. So, and wherever there is DNA, that's where the virus will head for. So nucleated cells, which are infected by viruses. Okay. So as we saw, when a cell is infected, a nucleated cell is infected by a virus, the nucleated cell secretes interferon. Interferon is a small protein. It doesn't disperse, too long distance. It's not like a hormone. So basically, it's in the everything is happening in the interstitial fluid. So we got nucleated healthy cells, which will absorb this interferon. When the interferon is absorbed by these healthy cells, which are not infected by the virus yet, but definitely they will be, because that's what viruses do. Infect a cell, make new viruses within that cell, then lyse the cell, rupture the cell from within. And then all the viruses are on lookout for new host cells. So today, tomorrow, day after, these healthy cells will be infected by this virus. That's a given. So this is why I was saying that the nucleated cell that secretes the interferon is like a whistleblower. It's blowing a whistle and saying that I am infected, save yourself. Here, this is what you save yourselves with, interferon. So these nucleated cells that get the dose of the interferon, remember that can the virus still enter these cells? Can a virus enter the cell that has already received the interferon?
Yes.
Yes. So viral entry will happen. The virus will enter, but can it replicate?
No.
No. So basically, what happens, the virus comes inside, can't replicate, it just stays within. And then as we'll see today, the same cell, which is now harboring a virus, and the virus has not replicated or hasn't been able to replicate yet, this cell is going to put the viral protein inserting into one of its own protein and call the different types of immunogenic cells toward itself. That will be the next step. So this is what I was saying, that innate immunity keeps the infection in check. Acquired immunity will wipe out this virus. But every cell that harbors the virus is going to do the same. They're going to insert a viral fragment, viral protein on its membrane, which is unseen. It makes it different from the self cells. And that's an open call to some of our, we'll learn it today, cytotoxic T cells or CD8 cells. So basically CD8 cells, which are a group of immunogenic cells, will be attracted to this. And along with this, CD8 cells will have another group of T cells, the helper T cells, Cd4 cells, that will be built up. There will be all sorts of complex interaction. But the end goal is going to be every cell that's infected with the virus, but the virus may not have replicated or may have even started replication, doesn't matter, is going to be wiped out by the adaptive immunity. So this is what I mean by innate often keeps the infection at bay. Adaptive wipes it out. Then complement protein. Oftentimes, understand that all of these different proteins and cells, they are designed for entities, maybe the same entity, same virus, at different places though. So complement protein has no way of detecting a virus that's inside the cell. A virus that has infected the host cell, complement protein will do nothing toward it. But complement protein will recognize a virus when it's traveling. So basically, the virus will have to travel from one cell to another, right? And that's where the complement protein will get it. Complement protein may come in contact with viral, traveling virus, or bacterium. Bacteria never invade into our cells. Bacteria are free living entities. So what the bacteria normally do, they colonize our interstitial fluid and they just start reproducing at an exponential rate, taking away our resources, which is why you often see one bacterial infection giving rise to another bacterial infection. You see that a serious infection can give rise to opportunistic infections. So basically it weakens our bodies, takes away resources. And then of course, extra cells, their products are toxic. So either a virus which is traveling, oftentimes you see this, that many students think, oh, viruses can't be free living, they're inside a cell. No, but a transmigrating virus, it's called a transmigrating virus. Traveling from one cell to another cell. Or bacterium. Bacterium are always is bacteria E or I? Gosh.
E.
Yes, I thought so. So basically, it's a transmigrating virus or a bacterium. Bacterium which never hides anywhere. So complement protein basically will recognize a pathogen which is outside in a body fluid, not within your cell, be it virus, be it bacteria. And as we saw, complement protein will get activated when it comes in contact with. The textbook talks about the classical pathway, alternate pathway. I usually say, leave that on the side. Know that complement protein is activated when there is a free living pathogen. And the way it was activated dictates what it's going to do. It's like a clotting almost. It's chain reaction. One protein interacting with another, then together they interact with the third. Like this, few complement proteins come together and they make a protein. That protein could be, as we saw, could be a MAC, membrane attack complex, which punch a hole, or it could be an opsonin, which will cover the entity, it is part of the entity, and makes it more visible to the phagocytes. Opsonins are those proteins that opsonize. The process is known as opsonization. Opsonization, like I use the analogy that someone in dark in your house, you do have a gun or you do have people who have ways to defend yourself. But it's dark, you can't even see. Phagocytes often will be like that. Oftentimes, phagocytes themselves will recognize, but it could be slightly delayed. But it's like in a dark room, somebody flashes a light right on the intruder and says, right there, makes them easy target. Now, whoever is defending you and your family and your house, they can immediately start attacking that intruder. So opsonin, opsonization, complement protein. I really don't know what to do with the Microsoft search. How do I deactivate this? Because this is annoying. Okay. Oh. Thank you. Then antimicrobial cells. It's so fascinating. This is part of our immunosurveillance. Basically, NK cells and ultimate, the king of innate immunity, phagocytes. And as you saw, NK cells travel. When you see immunosurveillance, what do you mean immunosurveillance? These cells don't stay put in one place. Unlike most of our immunogenic cells, the B cells, T cells, typically they stay put, except for one type of T cell that will travel. But these cells, NK cells and phagocytes, they're traveling at all times. They're basically looking for, where? Do you have something that doesn't belong here? And that something could be cancer cells, which NK cells will recognize. An NK cell attack is the same thing, punch a hole. NK cells produce a protein known as perforin. Once they have recognized that, this could be a transmigrating virus or it could be a bacteria, as long as it has a different membrane protein, NK cell will recognize it. The idea is it's all about protein, meaning these cells, they can't just secrete something from a distance. These cells have to bind to the pathogenic cell in order to destroy them. It's like, think about that, you are a security agent and you can't just shoot at someone. You actually have to grab the intruder. Yeah, there has to be contact. So this is why it's all about proteins. NK cells must be able to bind to the pathogenic protein. Once they bind, then only they release the perforin. So it's not like stray perforin molecule everywhere. Then, that could be harmful for our own body cells. Basically, the NK cell binds to the pathogenic cells and right then and there discharges the perforin. And that perforin is going to go straight into this pathogenic cell. And as we saw, phagocytes, all of this, complement activation, interferon action, NK cell killing cells, killing either cancer cell or pathogenic microbes. What happens to this dead cell then? This has to be removed too. All of this will attract phagocytes. This is why phagocytes, you can call them the king of the innate immunity. Ultimately, it's the job of the phagocytes to reach to the site where everything is going on and then basically eat dead cells, eat live bacterial cell or live viral cells, eat your cells that are dead. This is why inflammatory response, everything that innate immunity does, it culminates into basically all of the components of innate immunity. I won't say all, chemical and physical barriers don't, because those are barriers to entry. Innate immunity components, complement proteins, interferons, NK cells action, basically all of those end with inflammatory response. Which, as we saw, localized vasodilation, localized edema, interstitial clotting, then phagocyte migration, margination, and immigration, and then phagocytosis. Textbook talks about in details phagocytosis. I usually say forget about that. Unless you are going to go into that field of research, it's more critical. It doesn't really talk about the steps of the inflammatory response, but rather it talks about phagocytosis. I usually say just know the steps. It's better to know the steps of the inflammation than the steps of phagocytosis. Phagocytosis is phagocytosis. The phagocytes will phagocytize, meaning they will eat. So this is why all of this culminates into inflammatory response. You have complement action. It will make the inflammatory response stronger. You got NK cells killing. It will make the inflammatory response stronger. You got interferon. It will make the inflammatory response stronger. All of these components, basically, because the end game is phagocytes have to be there. So phagocytes must be attracted. So all of these other components will culminate into a response that will be an open call for the phagocytes. Come hither. And phagocytes are amazing cell. Once a phagocyte recognizes that this is the target, once it has recognized the target by the picking up the chemical trail, which is why the chemotaxis, I'm going to conflict with Tortora, I said this in the lecture, I'm going to conflict with the Tortora about where the chemotaxis begins. The Tortora, and I have double, triple, and quadruple checked. The Tortora mentions that the chemotaxis begins within the vessel. No, according to my research, based on everything, all the other books, not all the other, but I did not consult all the other books, but there are 3 core books that I always go back to. So basically, chemotaxis is actually happening in the interstitial fluid. So if I'm asking you where does chemotaxis begin, I would like to see the response of it's happening in the interstitial fluid, not within the blood vessel. So the way it works for phagocytes, I'm going to clear all the drawing. So when we say phagocyte migration, excuse me, talking about opportunistic infection, weakening for anything and there are opportunistic infections. So phagocyte migration is margination, slow rolling over camp. Then emigration. Then chemotaxis. Because chemotaxis is a special type of movement where the phagocyte actually plays with, manipulates chloride ions. And it does not happen within the blood vessels. Because within the blood vessels, we have a range of every ion that we maintain, which does not get disturbed in case of inflammation. But in interstitial fluid, you can take a little bit of liberty. So basically, when a phagocyte is outside the vessel, initially it almost, the phagocyte behaves like, I don't know where to go. Where am I? Which way? Which way? So it will turn almost sometimes 180 degrees, 360 degrees, looking for where is the trail coming from. And so dead cells, complement activation, interferon, all of those have already happened. And so there could be clotted bacteria here with some of your dead cells. And wherever those clotted bacteria are, this is why we localized and clot the bacteria. They are not spread out. Few clots, that's where you will find most of the bacteria. And every phagocyte will pick up that trail of either this or this, or this, whichever is closer. Once a phagocyte, let's say this phagocyte has picked up the chemical trail from here, then it has that chemotaxis. And I kind of like to imagine it as if you are on your elbow on the floor and you are approaching, something that's on the floor, how you'll move on your, you know what I'm saying? With your elbow, move your body inch by inch with your elbow. Phagocytes, almost that chemotaxis movement is like that. Little by little, it will move. It's slow but surefire, meaning this phagocyte will not lose track of its target unless it's killed. It will stay on this trail, completely not deviate. You want to deviate this phagocyte, you have to kill this phagocyte. Amazing. So this is what chemotaxis is, that surefire way. This, like a biologist term, but it's called a surefire way, meaning once you see that chemotaxis beginning, you know that this phagocyte is on trail and it's not going to deviate unless it's dead. And they approach here, phagocytize, bleach out everything. Isn't that amazing? Secrete a bleach-like substance, meaning not only everything is eaten, everything is cleaned, a bleach-like substance is released so that things are cleaned out. So these are the master cells of the innate immunity.
The book mentions residual bodies as being released at the end of phagocytosis. Are those ever able to be recycled back into the body?
Actually, they are. Residual bodies are nothing but broken down. And the new edition of the Totora is changing some of the language. I don't know why, but anyway, basically, the residual bodies are nothing, but they're mostly broken up proteins, broken up into amino acid. And so, yes, most of those are actually recyclable and reusable. Some of it is not, and that will have to be eliminated, but most of it is broken up protein.
Okay, yeah, the book mentions most of it could be broken down. The amount of residual bodies that could not be recycled, is that substantial at all? So could that actually have negative side effects?
Minuscule.
Okay.
Phagocytes have this amazing ability to basically, because phagocytes are full of lysosomes, and the lysosomal enzymes of phagocytes, each lysosome of the phagocyte has 40 different types of enzymes. So no matter what type of protein are you talking about, phagocyte has the ability to slice it into tripeptide, tetrapeptide, something like that.
Okay. And then just one more quick question. You'd mentioned about the NK cells using perforin as having to be within contacts, but with that distinction, are there other ones, I guess, other than maybe the interferons that are able to affect other cells at a distance?
At a distance, for innate immunity, no.
Okay.
For adaptive immunity,
because you made the distinction. I know phagocytosis kind of pretty much is the same thing. It needs to be in contact in order to really do anything.
Yeah. Yes. Okay. In adaptive immunity, we'll talk about cytokine. Cytokine is capable of, but in a distance, kind of gives this wrong idea. I feel that when you say distance, you're thinking of, you're talking about between arm and forearm. No. When you say distance, you're still talking about interstitial fluid, pretty much localized, and maybe it's not right next to the cell, but it's just few microns, distance away. That is distance for this kind of reactions. So anything that's non-contact is distance.
Right. That's more or less what I was getting at.
Yeah. So no. In adaptive immunity, cytokines, yes.
Okay, thank you.
Yeah. Also keep in mind, most of this is happening where? Within the lymphatic system. Not just any interstitial fluid anywhere, but most of it is happening in the lymphatic system. This is why that everything is connected. What did we learn about for the capillary exchange? That the excess fluid that's in the interstitial space is drained into the lymphatic system, remember? And the lymphatic capillaries are large and they have large gaps or clefts in their wall so that actually whole cells can be moved in and out. So most of the bacterial population that are in our interstitial fluid, a large percentage of them are actually entering the lymphatic system and passing through the lymphatic capillaries. Then that's why, most of these reactions, that different processes we are talking about, they're happening in different parts of the lymphatic system. Most of the phagocytosis is happening within the lymphatic nodes. This is why we got lymph nodes everywhere.
Professor, could you explain a little more detail how the chems work on the margination?
Okay, so the CAMs
to slow
basically,
down the phagocytes?
yeah, the selectin and integrin. Okay, let me bring up the slide
Thank you.
here. Just excuse me. So here. So basically, CAMs, where is the share? Excuse me. Hey, where is the whiteboard? Okay. Okay, so basically the CAMs, normally the CAMs are, think of the CAMs, if you guys can see my hand here, I'm in a different place today. Excuse me. Just a second. So CAMs are basically, think of CAMs like two cells. Connecting CAMs, think of it like this. See the interlocked fingers that are basically suppressed? These are not sticking out. They are
Yes.
actually, yes. This is how CAMs are normally. So in a blood vessel, think of the two endothelial cells and the CAMs between them like this. So selectin is a CAM. What it does, it becomes like this in case of inflammation. See, sticking out.
Okay.
So basically, what it does to the blood vessel wall, which is normally smooth, as we know, like a polished wood floor. From polished wood floor, it becomes patches of carpet wherever the selectins are sticking up.
Mm-hmm.
This causes a phagocyte to forces it to slow down.
It's like a
Same
little
happens with the lymphatic
obstacles.
vessels.
It's like little obstacles.
Yeah, basically, yes. Think of it like a skateboard. You're riding a skateboard. And let's say you are riding in your house, although you should not, but let's say you are. So you have polished wood floor and you are riding your skateboard nicely and your skateboard hits the carpet. Immediately the skateboard stops. You have to get off the skateboard. So similarly, phagocytes can just zoom past in a smooth vessel, be it blood vessel, be it lymphatic vessel. But if it hits a CAM, it has to slow down.
Mm-hmm.
So once the phagocyte slows down and goes from CAM to CAM, the phagocytic behavior changes. It goes from one CAM to another CAM, kind of like, a hound dog. You make it sniff blood, or any dog, and the dog will start sniffing the floor and the ground and move slowly, not running past.
Yeah.
That's what the phagocytic behavior becomes, as if it's sniffing.
And they get activated by the inflammation.
They get
of
activated
the
by the
area.
inflammation. It's basically the sniffing. So it sniff, 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 go to selectin. And here comes an integrin. So integrins are also normally like this, but in case of inflammation, this,
Oh, okay.
creating gap. So basically
They
every
pass
phagocyte,
through.
yes, that goes through selectin, snip, slow. Oh, here is an integrin. It will squeeze out, emigrate.
Okay.
Without the CAMs, the slowing down won't happen, which is selectin. Without the integrin, this emigration won't happen.
I see. Thank you.
Sure thing. Okay, so then, all right, so let's go over acquired immunity now. Let me stop this share and I'm going to start the new share of.
One more question before we go back. So whenever the inflammation is healed and it stops, so the phagocytes, they squeeze in again?
They do what? Yeah.
They squeeze in to the...
Not into the blood vessel. If it's a blood vessel, then no. Because, by the time the inflammation ends, the CAMs are back to where they were, but lymphatic vessel, yes.
Yeah, the capillaries of the lymphatic system. Okay. Thank you.
Does the flow of the lymphatic system all just go back to the subclavian vein?
Yes, yes,
Okay.
but before that, along the capillary, along the trunk, basically all these lymphatic nodes are there, which are packed with phagocytes and packed with the cells we are going to talk about now.
Okay, thank you.
So it's expected that what you're picking up in the interstitial fluid could be loaded with bacteria, but the fluid that's entering the subclavian vein at the end of the journey is free of most of that bacteria because all of that was taken care of through the lymphatic nodes.
I see. Thank you.
So acquired immunity or adaptive immunity. Okay. So as we know, it's specificity, meaning it's very specific. Although, innate immunity also, there is recognition of self and non-self, but the same phagocyte will phagocytize virus A, virus B, virus C, virus Y. If you have 4 different viruses or 100 different bacteria, same phagocyte will do the same to all of those. Phagocytize, 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 phagocytize. There is no specific way for bacterium 1, another for bacterium 2. That's what specificity really means. But adaptive immunity not only recognizes self and non-self, is highly specific in its response. Meaning, understand what this pathogen is, understand its protein, and come up with a counter protein. It's all about protein. So virus A or virus 1's protein and virus 2, they're 2 different proteins. Bacterium A, bacterium B, 2 different proteins. Adaptive immunity, you respond with like A for A, B for B, 1 for 1, 2 for 2. 4 different types of protein for 4 different types of pathogen. Although 2 of them are virus, 2 of them are bacteria, but it's a different bacteria. That's how highly specific it is, which is why basically protein against protein. This is the protein we're dealing with. Okay, let's activate a group of cells that can bind to this protein. So carried out and memory. You'll see how that memory happens. So carried out by lymphocytes, B and T cells, but phagocytes are like a foundation. This is a foundation. Everything that happened in the inflammatory response, culminating with phagocytes eating the virus or the bacteria, that's the start of the adaptive immunity's response. That's the first thing that has to happen. Phagocytes must eat and present to the immunogenic cell. This is all built on recognition of antigen, which is a name for a non-self protein. That non-self protein is often on the surface of the virus or the bacteria, all kinds of pathogen. So quickly, what is an antigen? Antigens are basically fragments of foreign material that triggers an immune response. So anything that can trigger an immune response in your body is considered an antigen. For most of us, that antigen is a protein, part of a viral body or part of a bacterial body. For most of us, this is what an antigen is. Like the show here. Think of it like a viral body, and it got this little tiny surface proteins. This is what will serve as the antigen, not even the whole protein, just the tip. If it's a hyperactive immune system, which is somewhat confused, it will think of pollen as a protein, dust as a protein, other things, food as a protein, and start reacting against it. And this is what allergy is. Hypersensitivity is called hypersensitivity. An overly sensitive immune system that's treating harmless things as antigens. So we'll talk about that in limited, because allergy itself will be like a full half a semester worth of lecture.
When you
So
said,
what is an antigen? Sorry?
when you said most of us, it's virus and bacteria, is it because of, I believe in that lecture that you posted that it was like parasites as well, right? For like if we lived in,
Parasites as well, yeah. It's
Okay.
because the parasitic infection is so minuscule compared to the viral and bacterial infection. I'm kind of focusing on virus and bacteria. But yes, parasites too.
Okay.
It's slightly different for parasites, but yeah, that is there too. So it could be microbe, parts of microbe, bacterial toxin, meaning it doesn't even have to be a surface protein, it could be a protein synthesized. And for hypersensitive individuals, it could be pollen, transplanted organs, which have a different surface protein, etc. So I just have the epitaph there. Don't worry about memorizing what it is. Okay, so then what is the self, non-self? What is, we're saying antigen, a non-self protein. What does it mean by self and non-self? All of our cells, we learned this in 144, have unique surface markers. These are part of what we call major histocompatibility complex. That's why it's a complex. It's not a single protein. It's a group of proteins together, known as MHC. There are two types of MHCs in everyone's body. MHC1, which are built into the cell membrane of all nucleated cell. This is why you don't see them on red blood cells. Red blood cell, you see the isoantigen. RBCs have their own surface marker, isoantigen, that dictates our blood grouping, remember? But all other cells, which are nucleated cells, will have the same MHC1. So I usually say, think of it like a tattoo. My name is Sheena. Let's say the tattoo S is drawn over my cells. Every nucleated cell in my body will show that tattoo S. And my tattoo might be like this. And then another cell, which has the same tattoo S, that S might be like this, which is different. That's radically different for our body's perspective. This and this, when we are reading, we are saying, we are thinking drastically different. No, all it needs is slightly different surface protein. That's enough for immunogenicity, meaning immune response to respond, immune system to respond. So all nucleated cells. MHC2 markers are on membrane of cells that we call antigen presenting cells. Antigen presenting cells are written as APC. What they do, antigen presenting, the name tells you what they do. Antigen presenting cells basically display an antigen by inserting with MHC2 marker, and basically it stimulates or it facilitates other immunogenic cells like B cells and T cells to recognize the antigen and start or help with an immune response. So basically adaptive immunity responses, what it will culminate into, unlike inflammatory response, this is what we refer to as immune response. So everything that we'll talk about today and next class, basically we're talking about immune response. All the interaction between different cells, presentation, etc., will culminate into what we call immune response. That's what adaptive immunity does. It comes up with an immune response. So starting with MHC1 and MHC2, this is what all of us will have in our cells. Nucleated cells will have MHC1. Antigen presenting cells are present on MHC2. And a large part of our antigen presenting cells, at least for the class, but we are talking about the immune system in a way, very compact way. Basically, macrophage. So the same macrophages that phagocytized, what we just saw, phagocytized the pathogen, are actually going to be inserting part of the pathogenic antigen into the MHC2 and Again, like I said, open call, come hither. Who matches with this? So I'm going to talk about that, the fundamental of this.
Professor, would that be the MHC? Would it be responsible for recognizing where like a bacteria can be or cannot be by the because like the surface markers, for example,
Mhm.
that's what I was gonna ask you. Like every node has like differentiated cells or no? For example, like intestinal nodes, would they have like a different cells that would recognize like the bacteria?
When you say cells, which cells are you referring to?
The
Immunogenic
lymphatic
cells?
cells, like
Okay, so
if,
I think I understand what you were saying. So basically, you are asking, will each lymphatic node have groups of different T and B cells?
yes.
Yes and no. Basically, not every lymphatic node is going to be packed with different groups of B and T cells.
Mm-hmm.
But what is going to have, it's more complex than, than the scope of the class allowed. There are cell lines of T and B
Mm-hmm.
cells. But basically, we'll have B cells and T cells. At least it's expected that there will be at least one or two of the B T cells that have the matching surface receptor that can bind to every bacterial or viral protein that, that we are exposed to.
Yeah.
If we are not exposed to it, then it's a novel infection, just like corona.
Yeah, because I was wondering like how some bacteria can live in our system, our body system, and be like not be attacked.
Because they are basically hiding within something. For example, the tuberculosis bacterium that
Mm-hmm.
lives within our body fluid, but it's hiding within the phagocytes in a phagocytic vesicle, phagosome. So
Okay.
when we have tuberculosis infection, the bacteria comes in and it penetrates the barrier, and yes, we'll have an inflammatory response. Phagocytes will phagocytize whatever number of bacteria there are. And then the phagocyte will carry the tuberculosis causing bacteria.
Mm-hmm.
I'm forgetting the name, Mycobacterium tuberculosis, I believe. So basically that bacteria will be within a vesicle, and it will stay contained within the vesicle, and a phagocyte will carry it. Eventually, these phagocytes die in action, combating one thing or the other. Eventually, when this phagocyte dies, another phagocytes will phagocytize the phagocyte and will inherit those vesicles with the mycobacteria. This is why it's said that once you have been exposed to tuberculosis, it can precipitate any time. Because it's almost like an opportunistic infection. If your immune system weakens, if your nutrient level weakens, if your overall bodily system weakens, then it can precipitate out a tuberculosis. But other than that, no. So basically, that's how a bacterium can hide within our body and not be attacked. It's hiding within a cell that's supposed to combat the infection within the phagocyte itself.
Okay, thank you.
Sure thing. Okay, so basically, again, understand the fundamental principle behind. I'm going to insert a slide here. It's not, it's not really that. Oh gosh, why does it do that? I want a blank slide. So basically, it's not complex once you understand what is this response of the adaptive immunity. Basically, the response of the adaptive immunity is all about protein. We got a novel protein, meaning a protein that we have not seen before. That novel protein could be on the surface of a virus, it could be on the surface of the bacteria, or it could be the protein discharged by a dead virus, dead bacteria. It doesn't matter. It's a novel protein. We have not seen it before. Now, we have these cells known as T cells. I'm going to talk about that in a second, just to give you the overarching idea. B cells. These T cells and B cells, there are different types of surface receptor. It's all about receptors. Recall, receptors are necessary to bind to anything. So the goal of the body in any kind of adaptive immune response is to look for which T cell, which B cell can bind to this novel protein, meaning which T cell, which B cell has the surface receptor. And typically we'll have at least one or two somewhere that can bind to this novel protein. It will have the receptor. It's all about shape. The goal of the adaptive immunity is to isolate that T cell, isolate that B cell, and clone, produce a large number of the same cell, clone it, so that now these clones are effective against this bacterium or this virus that initiated the cloning. If you have a second bacterium or a second virus which are different surface protein, different antigen, that is, these clones are completely ineffective. Then the same process begins. Where is the T cell, the B cell that can bind to this pathogen, this antigen, which has the matching receptor? Isolate, clone. So in order to isolate and clone, you have to first find out who can respond to this, right? Just imagine that, you know an intruder, an enemy of the state. I'm kind of using Hollywoody language. Enemy of the state. This is almost like a movie. If I had the money, I'll make like an immune response movie. Alas, I don't have the money. So basically, think about you got an enemy of the state, and the enemy of the state exists, but the guys that are, or the girls that are people that are going to be able to eventually kill that enemy or localize that enemy, keep them content. They don't know what the enemy looks like. And you don't know which one of them is capable of recognizing the enemy. So basically, what do? You have a picture of the enemy and say, who can recognize? Who can recognize? Who can recognize? Who can recognize? So this whole involvement of the phagocyte is basically in that process of first isolate the cell. So this is a sequential process. The sequences, I kind of came up with this, but it makes it easier to put the CMI and AMI response. So acquired immunity mounts a response against what it considers to be a non-self cell or protein, like I showed, either a whole cell or just the protein that's discharged by the pathogen. Based on identification of the antigens, it goes through the steps of presentation, recognition, proliferation, attack. Attack could be two different types, either attack to kill or attack to immobilize. And finally, memory. Now we got new clones of cells which are capable of identifying this same pathogen if it enters our body again 1 year later, 2 years later, 5 years later. Some of them live for decades. This is why you can take vaccine and be immune for a decade. Sometimes it's for life, because how long the clone cells will live. So we'll talk about that in a second. So let's go through this antigen presentation. Understand the steps, and then we'll put it together as CMI response. Okay, so antigen presentation. Antigen must be presented to the B or T cell. Like I said, B T cells, they have surface receptors. Goal is to identify which cell with a receptor can bind. And presenting is at two different levels based on who the pathogen is. Once the B or T cells recognize the foreign antigen, then these are the final cells that will start mounting, the response. T cells only recognize fragments of antigens that are presented as part of MHC1. And when an antigen is presented along with MHC1, it's called MHC1 antigen complex. Well, actually, they can recognize MHC2. I take that away. Let's just call it MHC, not MHC1. There are two types of T cells. Part of MHC. Basically, there is the MHC, the antigen has to be inserted. Then only T cells can bind, recognize and bind. B cells can directly bind either to the pathogenic antigen without the MHC or just the antigen, floating protein. B cell can bind to that. B cells can also bind to MHC antigen complex. B cells are more versatile. Now, where is that antigen then? Antigen can be in two different places. If it's a virus, antigen is inside the cell. If it's a bacterium, then antigen is outside in the interstitial fluid. So the presentation is done by two different entities. One is your nucleated cell, another is phagocytes. So antigen in the body fluid presented by cells that we just saw, antigen presenting cells. A macrophage that phagocytizes. In inflammatory response, we saw macrophages phagocytize. Once they phagocytize, macrophages will basically insert the antigen with their MHC2 and display as MHC2 antigen complex. No, this is not this, sorry. Here. Yes, MHC2 antigen complex. So basically, surface protein along with the antigen. See that? That pink part, basically that's the antigen. The green part is the MHC2, which is always present. So if this is a macrophage, it always has the MHC2. Let's say this is what the MHC2 looks like. Now, it ingested a bacterium or transmigrating virus, inserted the antigen, bacterial or viral antigen, and now this becomes MHC2 antigen complex. This is, I have it written somewhere. MHC2 antigen complex instead of just MHC2. What about the virus? Well, virus infects our nucleated cell, as we saw. So when the cell is secreting interferon, it's also doing something else. Or those cells that are infected with the virus and the virus has not replicated yet, they do the same thing. They got MHC1. This is MHC1, normally present on surface of every single nucleated cell. But now that MHC1 gets an antigen fragment, and now you call this MHC1 antigen complex. So we're just covering the foundation, then I'm going to put the process together. Then presentation, recognition. Recognition will be whichever B cell, whichever T cell can recognize this, meaning can bind. It's all about binding. At least one or two, somewhere, T cells will recognize MHC1. At least one or two other types of T cells and B cells will recognize MHC2 antigen complex. So MHC1 with antigen, MHC2 with antigen, some cells, some immunogenic cells, somewhere will recognize this. And there are two groups. Those that bind to MHC2 are not the same as those that bind to MHC1. However, whichever cells recognize and bind either with MHC2 antigen complex or with MHC1 antigen complex, two different cells bind to these two types of antigen being presented, they will require basically B and T cells. They would require a chemical, small chemical, which is like a hormone known as cytokine. Cytokines, the S is basically there are a number of cytokines that are present. And this cytokine will come at a specific time of this process so that the cells can be cloned. And the cytokine is secreted by some T cells and antigen presenting cells. And you will see how that means. Right now, like I said, I'm covering just the basics. Typically after antigen recognition, meaning let's say this is a macrophage. This is displaying an MHC2 antigen complex. I'm just going to write it as MHC2 antigen complex. Once a matching cell, meaning a cell with matching receptor, binds to this, then this APC itself will secrete the cytokine. Put that together. Let's talk about CMI response, cell-mediated immune response. Okay, I think I'll go at least 5 minutes over time. So basically, CMI response, which is very specific, typically specific to viruses. Begins with activation of T cell by a specific antigen. So I'm just going to draw this in a second. Okay, result is a T cell capable of an immune attack. And this is the sequence again: presentation, recognition, proliferation, attack, memory. Next question is then, how many types of T cells are there? Are all T cells the same that are involved in immune response, CMI response? No. We got overall 4 types of T cells. Cd4, known as helper T cells. Sometimes they are written as TH, helper T. CD8, this is the name of the receptor, CD4 protein, CD8 protein. Memory T cells, which could be clone of either CD4 or CD8. And suppressor T cell, this is not discussed as much, CD25. So they are not as well understood. So CD4 cells, these two, CD4 and CD8, is what our focus is. CD4 cells recognize MHC2. These cells recognize MHC2 antigen complex, wherever that MHC2 antigen complex is. And we just learned MHC2 molecules are on antigen presenting cells. So when antigen presenting cells, like a macrophage, after it has ingested a pathogen, when it inserts the antigen along with this MHC2 and displays which group of cell is going to try and bind and be a match, CD4. B cells can do the same, but let's focus on CD4 right now. CD8 cells, they recognize MHC1 antigen complex. So when a nucleated cell is infected with the virus and is displaying the viral antigen with its MHC1, who is going to recognize this? CD8, not CD4, not B, CD8. CD8s are powerful killer cells. So as you will see together, put it together in a second. So they require 2 sequences of activation. Basically, they recognize the MHC1 antigen complex. Then they have to have the cytokine to proliferate. And even after proliferation, they require what we call co-stimulation, meaning help of a CD4. They require 2 binding. Bind with the MHC1, detach, clone, and co-stimulation, bind with the CD4 clone, then become fully active because these are killer cells. They will kill your body's infected cells. So they require two stimulation. One is binding, second is co-stimulation. And both CD4 and CD8s, when they clone, certain percentage of the clone will stay in our system as memory cell in case of a second exposure. So I'm going to quickly go through this. Then we'll have a review next class because I do want to finish my response by next class. So presentation, recognition, proliferation. So I'm going to draw this here. So this is specific to viruses. So let's say virus 1. This is what it looks like. Entered your system. Where would viruses go? To your nucleated cells, host cells. So this is your nucleated cell. Virus enters the cell. So you got viral infection. Next, this cell, which is infected by the virus, is going to display the MHC1 antigen complex. So let's say that's the MHC1. That's what it had before, just MHC1. Now it's going to have MHC1 with the antigen. So this is just MHC1. This is MHC1 antigen complex. Now CD8 cells. These are CD8 cells with matching receptor will recognize. So this is presentation. Display, this is presentation. Now the CD8 cells with matching receptors will try to bind to this MHC1 antigen complex. That's what they recognize. It's almost like one cell comes in, tries to bind, not a match. Okay, go. Another, no, not a match. Third one, yes, match. So the recognition is cells trying to bind, and eventually we have at least one, like I said, at least one CD8 cell or two CD8 cells somewhere. This is the first infection, which has the matching receptor. So recognition. Now we got the nucleated cell with its MHC1 antigen complex, and we got a CD8 cell that binds. This CD8 cell will next need to proliferate. Okay. So while this is going on, simultaneously or pretty much ahead of time, inflammatory response already happened, right? So you can say like, meanwhile, it's like a story. A macrophage, which has phagocytized the same virus. The virus has entered, it has already killed, while this is going on, it has killed few cells and transmigrated, that's the idea. And a macrophage has ingested the same virus. The macrophage will now start displaying MHC2 with the same antigen. So MHC2 antigen complex. So that is their presentation. So we have two presentations going on. One for viral infection, one at the level of the nucleated cell, one by the macrophage. So as soon as the macrophage displays this MHC2 antigen complex, this becomes an APC. Now this is an antigen presenting cell. It's presenting the antigen. Who would, same thing, one by one, Cd4 cells will try to bind to this, because that's what they recognize, MHC2 with antigen. One by one, Cd4 cells try to bind. No, not a match. No, not a match. No, not a match. Yes, match. And now the CD4 cell recognizes and with the matching receptor binds. We got at least 1 or 2 CD4 cells that will recognize this antigen with MHC2. Now the proliferation. While the CD4 cell is still attached to the APC will secrete a chemical known as interleukin 1. So I'm going to continue on. I know I'm going over time. I want to quickly do this. If you have to leave, you may leave, but I do want to finish it here on the go and review it. So MHC2, antigen complex, CD4, this APC starts secreting a cytokine known as interleukin 1. This is a cytokine. Who is it meant for? CD4. So interleukin 1 molecules taken by CD4. CD4 will detach and clone, proliferate. So while we have this nucleated cell with the CD8 waiting, basically these CD4 clones will travel, and wherever they find the CD8, they would bind to the CD8. Two things can happen. They would bind to the CD8 and secrete another cytokine, IL-2, simultaneously, or the CD4 will produce the cytokine, the IL-2, almost immediately. It's also possible that the IL-2 molecule will be reaching the CD8 cell first before the clone can get there. In that case, the CD8 cell will detach, clone. Each clone must shortly bind to the CD4, that is the co-stimulation, before the CD8 can become active, fully active. So this is what I mean by two exposure. First, bind to the MHC1. Next, bind to the CD4 clone. This part, I think the textbook kind of doesn't emphasize. It makes it sound like interleukin 2 is the co-stimulation. It's not. CD4 clones must briefly bind to the CD8 clone. That's what fully activates it, because these are killer cells. So it can happen at the same time, CD4 binding to the CD8 and discharging interleukin 2. Or it could be that interleukin molecules are discharged by these clones. CD8 gets that first and proliferates. And now the CD4 clone and CD8 clone will have to briefly bind. One way or the other, after the co-stimulation, you got a fully active CD8 cell. I think this is the first part that I was showing. A fully active CD8 cell. This CD8 cell basically will find every infected cell infected with the same virus. All of them have the same antigen in their MHC1. The CD8 clones will bind to the MHC1 antigen complex and kill this nucleated cell which is infected by virus. So this is what I was saying earlier, that your infected cells that are infected with virus, will have to be killed. Whether the virus is replicating in there or not doesn't matter. It's harboring a virus. It has to be killed. Who's going to do that killing? CD8. It's killing your own cell, which is why it requires two sequence, two binding. One, first with the MHC1 and antigen complex, second with the CD4 clone, which is called co-stimulation. And that co-stimulation can happen simultaneously with the interleukin 2, while the CD4 clone binds to the CD8 clone, and CD4 discharges interleukin 2, and then CD8 detaches from both and clones, is fully active then and there. Or it could be CD8 clones that are not fully active yet. CD4 clones will have to briefly bind. That is co-stimulation, not just the interleukin, binding by the CD4. It's like confirming that Yes, I can kill. Now the CD8 cell will do what it does, kill infected cell. Occasionally, they also recognize cancer cells. So this is an electron microscope picture that shows the CD8 cell and a cancer cell. This is the hole that the CD8 cell punched. So presentation, if you remember it like that, presentation, recognition, proliferation. Recognition goes with activation, basically. Proliferation, attack, memory. Now you got memory CD4 and memory CD8 and memory CD4. In case of future attack, you don't have to start with 1 or 2 cells. You got hundreds, sometimes thousands to start with. Unlike the very first exposure to this viral protein. You started with 1 or 2, maybe, 3 or 4, minuscule quantity. It's a novel infection for you. After your body is done with the first exposure, you got thousands of clones. That same infection, same virus appears again, you start with thousands of clones. So oftentimes you won't get sick. Before you get sick, the virus is overpowered by all these clones. The more you are exposed to that same virus, the more clonal cells you'll have. You'll have a larger stock of the clonal cell. At some point, they'll be so large that you are practically immune to this virus. That's why it's called adaptive immunity.
Professor, but why, okay, so the memory works like that, but why some infections or viruses, if you get for the second time, it can be lethal or it can be even worse?
It can be lethal or worse. Which condition, which virus are you talking about?
Like, for example, chicken pox. Like,
Yeah,
usually you get once, you don't get the second time because you already have like your immune system has a memory. But if you do get a second time, which is possible, then it can be like even worse than the first one, no?
It can be worse, but not necessarily lethal.
Oh, not in this case.
But typically second exposure to a virus or bacterium and that exposure being lethal is extremely
like tuberculosis, for example. Tuberculosis?
No.
It can be worse than the second time too, no?
No, not necessarily.
Okay.
Not necessarily. For tuberculosis, for tuberculosis, because the first set of bacterium has never died to begin with. If you're exposed to the second set, literally means your immune system gets more and more compromised. With successive exposure, your immune system is more and more compromised. You got more and more phagocytes with the tuberculosis, carrying the tuberculosis bacteria. So the risk of actually having tuberculosis increases. It's not worse. You don't get you may get the disease tuberculosis in first exposure, you may not. Oftentimes, if you don't have a weakened system, you don't actually have the disease tuberculosis. You don't see the bacteria induced damage in the lungs or anywhere. But yes, you are carrying that bacterium. Because you are carrying that bacterium, you have been exposed to that bacterium, you will have the antibody to that. So you do have some clonal cells. The second exposure literally means a further weakening of your system. So successive exposure to tuberculosis, what happens is that your stock of tuberculosis bacterium in your phagocytes increases. And because the infection was never nullified, you keep building up antibody to that tuberculosis bacteria. And if the phagocytes end up dying and the tuberculosis bacterium being discharged all at the same time, you got 100,000 of tuberculosis bacterium put away safely in your phagocytes and they are discharged and you got the antibody, you'll have a massive immune response. So the symptoms are much worse, not necessarily lethal.
Okay.
For other chickenpox, basically a second chickenpox infection literally means your memory cells could not be kept. So the same idea that
Oh, okay.
typically memory cells against the chickenpox is for, basically it's a vaccination process that most of us are exposed to chickenpox because of vaccination to the smallpox. Said that if you have a chickenpox, actual chickenpox infection, means your clonal cell stock either has been wiped out or is ineffective, which makes you at risk for smallpox now. These are deviation though. These are not what happens every day. If we live for there is this other thing also, the average life expectancy. The vaccine to the smallpox by exposure to chickenpox was developed way back when the average life expectancy was much less than what we have now. So the memory cells, the clonal cells, their life expectancy also was matched based on when we lived. So it was expected that you'll die by the time you're 65. So you get immunized to smallpox now, and we are good for next 50 years. But now you are 90 years old and your clonal cells did not survive that long. You need a second vaccination with the chickenpox again, but your immune system is not at the same level where it was before. And there are myriad of factors. But the statement that it's worse, not necessarily. For some individual, depending on where you are, like in a geriatric population, weakened immune system, you got a chickenpox infection, actual infection, which means your vaccination, if you are vaccinated before, basically has failed, or your first exposure, the clonal cells did not make it. And it happens in after a very long gap.
Okay. And like, for example, the flu, it's because the virus is always mutating, right?
Exactly. But flu is different. It's a novel virus each time.
Okay. Thank you.
Sure thing. Any other question?
Just kind of like a fun hypothetical. So as far as things like the flu that mutate, do memory cells ever interfere with one another in response to a new mutation?
Do memory cells, what do you mean by interfere with one another?
I don't know. Just the presence of previously created memory cells, do they ever cause problems with the body's response to a new mutated version, let's say, of the flu virus?
Interesting question. You know what? I don't know. Theoretically, no, but immune system, I would like to look that up. No, because immune system, there's so much research and new data coming in.
Right.
I wonder. I don't know. But
Yeah, I was curious because they would
things
be
stand,
like so similar, kind of.
as things stand, no. Theoretically, that's what I'm saying. Theoretically, no, because we are talking about even minutely different, but it's a different protein. So clonal cells that are against this protein typically do not interfere with something with this protein.
Right.
So hypothetically or theoretically, no, shouldn't. But immune system, the interaction between different cells, and more than that, also the interaction of our immune system with our intestinal bacteria, that part is very mysterious. So there are actually a lot of new data that's happening. This is the field of research for last 10 years, because cell and molecular biology is almost saturated.
Right.
Immunology is the new frontier. So I would, let me research, let me look that up a little bit.
Awesome.
Oh, I can see your dog in the back.
Yeah, I know, because I
So
put him out.
cute.
Let me stop recording for this.